Okay, on this page, when you're reading it, you want to make sure that you outline uh, what information was known before the experiment was conducted, and what was being used in the experiment. So when we look at why would we study pneumonia, and you look at questions like uh, what does it mean to grow on a plate, or you see that the pathogenic strains were shiny and smooth, and what does the word pathogenic mean? And when you read this section, it tells you a lot. This paragraph tells you a great deal about why they do the experiment the way they do it. So the word pathogenic means uh, disease causing. Right? Not all bacteria cause disease, but some do. Um, when you grow a bacteria in a plate, which we'll be doing right after the OSTs, you you wipe on an agarose gel, and everywhere there's a bacteria, a small colony will grow. And you'll be able to see, you can't see one bacteria with your naked eye, but you can see a lot of bacteria together, a colony of them you can see. So each one of these is known as a plaque or a colony of bacteria. That's why the plaque on your teeth, when you say plaque on your teeth, they're actually a colony of bacteria growing. So read this section, understand. Do you need to know Griffith? No, you don't need to remember Griffith, but it'd probably be a good idea to understand who's doing the experiment. So Griffith, and he's using mice. Why did he use mice why would he use a mouse as a model organism? If you remember, a mouse is a mammal. You remember what remember that a mouse is it's a mammal. It's very much like a human. They're cheap to raise. So there's a whole list, a bullet point list of why they're really a great model organism. And so you can, and of course this is the 1920s. If you look at the time, this is 1920s. They didn't know a lot about DNA yet. So when you're looking at why would we use a mouse as a model organism, again, it's cost, it's cheap, they have fast generations, they produce a lot of offspring, and we understand their physiology. So they made a good. That's why they used mice to study these. So read the paragraph, understand why that'll help you understand why they do what they do. If you jump right into the experiment, you really won't understand what's going on. So assuming you listened and you went through and you understood and you made a list, what was the difference between the smooth? The smooth did something. There's something about the smooth. The rough did something. Uh, etc. But to give you some background on the concept, so you should have a list from the previous page on what they thought was going on with the smooth and the rough. You see they're injecting each of these. In the first group, they inject with the rough strain. In the second one, they inject with the smooth strain. Then he killed the smooth strain, and here they put this rough strain with the heat killed smooth strain. So let's discuss for a minute what the background information is. So the idea is you have these cells. The rough cells they're rough because they have proteins that are on the outside of the cell. They don't have a smooth mucus coating. The other cells, the smooth cells, don't have the proteins, or the proteins are not visible because they have a thick layer of mucus of non-specific material that surrounds them, encapsulates them, puts them in a capsule. They do have proteins, but the proteins are here on the surface. And you remember with the ABO blood groups, the cell surface proteins allowed your immune system to tell you if you have, and let's go back and review ABO blood groups. You have an A blood group if you have A surface protein A on the outside of your body. It's not a letter, but I'm just going to put A. Your B blood group if you have a B protein, cell surface protein. Remember there's two genes, there's the A and the B. Remember they're codominant, so if you have one A from mom and a B from dad, then you have both A, B, so your AB blood group, blood type. And if you're O blood type, you have the recessive gene. That means that you don't have any surface protein. So that was the ABO blood group. That's human cells with ABO, A, B, or O genotype. And that was an example, by the way, of codominance. Now, when we're dealing with bacteria, though, this type of bacteria specifically had the rough strain, the protein is visible, so your immune system can say, hey, that doesn't belong to us. It's not A, it's not B, it's not O, it's something different. Kill this. So these bacteria, your immune system kill quickly. Because they're able to read the bacteria to see that don't belong to you, and so they die quickly. These, however, have this protective coating that confuses the camouflage. So the mucus layer camouflages the bacteria, and it doesn't allow for your immune system to react to the bacteria. So the bacteria grows and ends up, in this case, killing the mouse, right? Or not killing the mouse. That's the thought. That's the hypothesis that the smooth coated bacteria, they're camouflaged with that coat of material. So in this example here, they're going to inject the rough strain. They expect the expectation. Let me write down the predicted. Sorry, it already says. So this says predicting, right? So predict what you think is going to happen if you inject the mouse with the rough strain. Is it going to be is it going to live, or is it going to die? Well, if this is true, wait a second and see if you answer. If this is true, that what we think is happening, then we'd expect that, that something here is going to happen. You can write it in. The smooth strain, is it going to live or die? So we're pretty sure we know what's going to happen. We can easily predict what's going to happen here. So with the rough strain, if what we're saying is correct, this is going to live. If we're correct, the smooth strain, the mouse is going to die. Pretty straightforward. Here we have heat-killed smooth strain. So we're going to take this smooth strain bacteria that causes disease. We're going to kill it with heat. We're going to boil it and sanitize it. We're going to take the pieces and parts, the cooked bacteria, and we're going to inject it into the mouse. And do we expect it to live or die? Let's say we expect it to live. We're predicting it's going to live because the bacteria can't grow, right? That's our hypothesis, that this smooth coated bacteria is going to grow and kill the beast, the mouse. But if it's dead, it can't grow, so it can't kill anything. So the mouse should be alive. And then what's going to happen if I take the dead things and mix it with the live rough one? What do we think is going to happen? Since the rough doesn't cause disease by itself, we think it's going to live with rough bacteria. We think it's going to live with heat killed. Then logically, you'd assume it's going to live if you mix the two together and put them in. So then it comes another question as far as experimental design goes. Why do we do these different experiments, right? If we know that rough killed strain doesn't kill the mouse normally, why are we injecting the mouse with rough killed strain? Well, this is a negative control. We know it's not going to kill. We know the answer is going to be no, but we're going to do it anyways to make sure that it's not our needle, it's not the liquid that we're putting the bacteria in. There's no confounding factor that's going to cause this mouse to die if it does die. So we expect it to live. If it doesn't live, then that checks us. We're using it for comparisons, our control group, specifically our negative control. If we know that the smooth bacteria causes death, why are we injecting these poor little mice with a bacteria we know with something that causes them to die? Again, it's our positive control. We know that if it's going to die, it should die in this way. It should take this long. And it is because if it doesn't kill the mouse, then we have to wonder what's going on. Something else is going on. So again, it's our control group. We're using it for comparison. This is our first experimental group. He killed smooth strain. We expect it to live, but it's our experimental group. And so is this one. We don't know what's going to happen. Actually, it's probably fair to say that this is also a negative control. Because we expect that if it's dead, it's not going to cause death. But what happens when we mix them together? Now, why are we mixing them together is another big question, which brings us to the whole purpose of the experiment. The idea is that this is before we understood that DNA was the stuff of inheritance, before we understood what chromosomes were, before any of that. We know that this had a number of traits, some kind of trait, in this case, the mucus layer, that allowed it to be deadly. This was missing the trait. So the question is it possible that this bacteria could somehow give something to this bacteria that could transform it? Transform this bacteria from a non-killer to a killer, from not having the mucus camouflage to having the mucus camouflage. It's the material that causes transformation would be the stuff of inheritance. So they're looking for the stuff that you inherit from your parents, the stuff that life passes from one generation to the other to allow traits to transform a creature, right? So this is a transformation of bacteria. That's the thought. Let's see what happens. So they do the experiment. The rough mouse lives, check. So we're good with that. We expected that. The smooth strain dies. Positive control is correct. We're good. The heat killed one lives. Again, we expected that. This was a question now. Why is it that this particular mouse died? If it was injected with two things, it wouldn't kill it. If neither one of these things alone would kill it, what allowed this rough bacteria now? Remember, these are dead. We know this stuff doesn't kill them. We know the rough by itself doesn't kill them. What allowed this rough bacteria to kill the mouse? Well, the thought is that something in the dead material went into the rough bacteria and changed them, transformed them into this deadly form of bacteria. It gave the deadly trait to the non-deadly bacteria. It transformed the non-deadly bacteria to the deadly bacteria, into a deadly bacteria. So that's the thinking, right? So then you have to consider then in this experimental design, what would that material be? Well, we don't know, but we do know we do know something, and what we do know is that in that bacteria, in this dead material, are proteins, are lipids, are nucleic acids, like DNA and RNA. And we know there's carbohydrates. So we know those four things are in that bacteria. Today we know DNA, it's probably DNA and RNA, but how did they know it was? In fact, what they were pretty sure that it was proteins. This is what they thought. They supposed it was proteins because what they were thinking of is, geez, there's 20 amino acids, and if you're going to write this really complicated book of life, wouldn't it be nice to have 20 letters? Where in DNA and RNA, you only have 4 letters, 4 different DNA, and you only have 4 different RNA. So you only have 4 letters to write a really long code. It would take billions of more material to write in DNA or RNA than it would for a protein, because you can't. Proteins have 20 letters, so they can be very specific. Where DNA and RNA would take more. The molecule would have to be bigger to write the same message. All right. Okay. Well, they thought that. Today we know that that's wrong. Just to want to make sure you're not. We are clear that the stuff that's inherited is DNA and RNA. But again, a lot of people thought it was proteins. So let's go on and see how we know it's DNA and RNA, not proteins. So here they ask you to write down these are ACEs, so they are enzymes. What do each of them do? Here I'm going to let you figure this one out, figure out what each one does. I'll give you a few more seconds to think about it. All right, so proteases, they destroy proteins. They are enzymes that catalyze the breakdown or the hydrolysis, that means the breaking up of proteins. So they destroy proteins. So you put the protein in with this enzyme, proteins are destroyed. And DNases do the same thing. They catalyze the hydrolysis of DNA. And RNases do the same thing. They catalyze the destruction or hydrolysis of RNA. These are important tools, these enzymes. Without them, DNA and RNA and proteins would last a very long time. So if you have these three tools and you have a test tube that's full of this material, you have several of these test tubes. You have several of these test tubes, and you want to know, is there DNA? Is there RNA? Are there proteins in these? Right? Or if you want to get rid, if you want to make sure that you, if you want, I'm sorry, I'm having trouble with this. Oh, there you go. If you want to make sure that whatever's in this material is, whatever's in this test tube is not DNA, what would you do? Well, you'd probably add a DNase to the test tube. If you add DNase to that test tube, it gets rid of the DNA. Then only the test tube that's left is the same test tube, but there's no DNA. And the same thing's going to happen if you use the same sample, but this time you put an RNase. So that test tube, after you add the RNase, has no RNA. And then lastly, you would do the same experiment. This time you'd add a protease. Pretty straightforward. So now, when you kill that smooth strain bacteria and you put that DNase in there, and then you put it with the rough bacteria, if it transforms, then it's not DNA, right? Let's go and look and see what happens. So he killed the bacteria, then they add RNase. They add that RNase with he killed bacteria to the rough strain. The bacteria dies. Well, that means something transformed. That means something gave the trait to this rough bacteria. The smooth bacteria, the message got to the rough bacteria. So we know it wasn't RNA that was giving that message in this case to the bacteria. What didn't allow for it to be inherited from one generation to the next, that message, that inheritable message, was not RNA. How do I know that? Because I got rid of RNA. There was no RNA, and the message went through. If it was RNA, then the message would not have gotten through, and the mouse would have lived. So here's a smooth. He killed smooth bacteria. This time I had protease, so I got rid of all the proteins. It has the RNA, yes, DNA, yes, but no protein. Here I had protein, yes, DNA, yes, but no RNA. And still the mouse died, so the message still got through. So I know that the messenger was not a protein. And with this one, I know the messenger, the inheritable message was not written in RNA. But now I get rid of DNA, all the DNA in the test tube from the heat killed smooth bacteria. It has RNA, it has protein. I add it to the rough strain, mix it up, let the message get inherited. And then I inject that mouse with that mixture, and the mouse lives. I got rid of the message. It's not being inherited anymore. This was a really beautiful experiment to let us know that DNA was the inheritable material. So this was a very important experiment. The Mises install experiment on whether DNA. There were three ideas of how DNA is being replicated. It could have been that it was semi-conservative. In other words, the DNA strand is opened up, one strand is used to read, a new one is made slowly on both strands. At the end, you have two new strands, one old version and one new version. Here in the conservative uh, model, this is another way it could have been copied, is that the DNA molecules could have been opened up and then both strands were read, and then you had to end up with one DNA strand that had the two old strands of DNA and the new strand that had both new strands. So in other words, the old strands stay together and then two new ones stay together. And the dispersive model said, look, what if part of it was stayed old and part of it was new, somehow was pieced together like Frankenstein? Why does it have to be either one or the other? So those were the that three thoughts and three hypotheses that were being tested. Which one of these is the way DNA is replicated? To be clear, this is the way. It is semi-conservative. It is not the conservative model. It's not the dispersive model. But the point here is, how do we know that? So let's go ahead and figure this out. So you have nitrogen. You have different isotopes. And we know that nitrogen with, remember, isotopes are the number of protons are consistent. They don't change. They define the element. The number of neutrons, however, can change. Number of neutrons can change. And if you have a different number of neutrons, the number of protons, then you have a mass number that's different. So some isotopes are heavier than others. In the case of nitrogen, 15 is more dense than nitrogen 14. Now, if you paid attention when you're washing clothes, you know that when you spin the clothes in the spin cycle, the clothes stick to the side of the washing machine. But interestingly, what we can do with that centrifugal force is we can put it in something called a centrifuge, which again we will do right after the test. You can put these test tubes, you've probably seen them in TV shows, in these little test tubes, and they'll stick out and they'll spin from a rig that's in the center. And usually there's four of them going out. And when you spin them, all right, so if we took this material and we put some material in these test tubes, and some of it was, let's just make, let's make N15 red, and we can make N14 blue. If we did that, and they're together when we start spinning them, they're together. And let's say they're at the top. Usually you don't do all four, you just put some comment. Obviously you're going to want the same amount of mass in all four, so that they're balanced. You know what happens when a washing machine's not balanced. So you're going to spin them in a circle very quickly. And the faster you spin, the faster they separate. What's going to happen if we just look at one of these test tubes? What's going to happen over time is that the heavier one will be at the bottom and the lighter one will be at the top. So centrifugation, remember when we talked about gel electrophoresis is a way of separating things by molecular mass or by charge. Here we're able to separate things by density, and we call this a centrifuge. So, uh, so there it is. That's kind of the basis. This is the tool that we're going to use to study this process of how DNA is copied. So a good question comes up before we even get into the actual experiment is why nitrogen? Why is nitrogen so important? And I think if everyone remembers, when you look at a nitrogenous base, at a nucleotide, DNA and RNA both have this carbohydrate, this 5-carbon carbohydrate, either ribose or deoxyribose, 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. We've talked about that. The DNA and RNA both have this phosphate group sticking off the 5 prime. And DNA and RNA both have a nitrogenous base. In other words, there's nitrogen involved in this either single or double ring structure, right? And there's this, in this 1 or 2 ring structure, you have a nitrogenous base. So there's nitrogen in DNA, is the point. All right, let's go on and see what the experiment's like. So here, you see after growing the bacteria, nitrogen 15, producing heavy chromosomes. So all the chromosomes, so if we look at a chromosome with nitrogen 15, all the chromosomes, it would be all condensed, right? And they'd be copied. So these chromosomes are heavy because they're using nitrogen 15. And we could switch them over. So after you make copies of the bacteria, I mean a lot of copies, you switch them over to, so you make there's a lot of mitosis happening here, right? A lot of copying of DNA in S phase. During S phase of the cell cycle. Instead of mitosis, why don't we just say the cell cycle? Because remember, DNA is copied during S phase of the cell cycle, not during mitosis. Mitosis is separation of the nucleus. So after some time, we switch it over to nitrogen 14. If we did that, and so you have some nitrogen, it was heavy, and now it's not heavy. Red in this case is going to be the lighter version. And we spun it in that centrifuge, and we looked at the test tube. What we would find is we would not find the band. We'd not find the band at the lighter depth, right, where we would expect it. No band. Nor would we find it at the heavier site. So there'd be no DNA there either. Where we'd find it is somewhere in between here. There would be one band in the center. You see, there it is. 100% of the DNA is found there. So if that happens, if we grew DNA, we grew the bacteria in nitrogen 15, and if we stopped there, we would expect there'd be a band here because all the DNA would be here. We got rid of all the proteins, all the carbohydrates, all the RNA. There's nothing but DNA in this experiment as far as we're concerned. And then we grew it in nitrogen 14. What would the semi-conservative Could someone close the door, please? Model say. In the semi-conservative model, what we would expect in a semi-conservative model is there'd be a band in the middle, right? Because half the DNA would be light and the other half would be heavy. So there wouldn't be a band at the light or the heavy because there's half and half. So it'd be somewhere in between. All right. What about the conservative model, though? What the conservative model say is that you would have the old strand, which is made in this case of nitrogen 15. You'd have the old strand, the original DNA, before you switch to nitrogen, would be used as a template. Both the old ones would be kept together, and then both the new ones, which now are being made with nitrogen 14, would be kept together. So here, if this was truly conservative, if the conservative model were true, then what we would expect is there would be a band at the lighter region of the test tube and one at the heavier region of the test tube, because there's one chromosome that's made of nothing but heavy DNA and the other one's made of nothing but light DNA. So I'd expect two bands. So that would be conservative. So that one's not true because this is what we got so far. This is possible. This cannot be, because if it were true that conservative were true, then we would see two bands. We only see one in the middle in between heavy and light. So it's not this one. So we know at this point it is not conservative. Lastly, we have the dispersive model. Now in the dispersive model, it says that we grew it in nitrogen 15, so there'd be heavy, and then we used part of that as a template, and we interspersed it with some of the new. So there's some old and some new mixed in. So there's some old and some new mixed in. Dispersed within the DNA is some old and some new DNA. So if that were true, then we would expect that there would be a band in the middle. There wouldn't be two bands because they'd have the same molecular weight, or same density. So we'd expect to see one band in the middle. So this is not ruled out as a possibility. Only the conservative model is ruled out. So how can we figure out whether it's semi-conservative or whether it's dispersive? I want to remind you again, for the sake of your test, that semi-conservative is correct. That is how we do it. But we still don't have the empirical evidence, the experimental evidence to support that concept. Well, let's go on and see what they did. So what they did is they ran it through the first generation. So they had the bacteria. It was on a plate, a bunch of bacteria on a plate. They grew it in nitrogen. They grew it in nitrogen 15, right? So all the DNA, they put in a test tube. They got rid of all the other stuff. The only stuff in the test tube is DNA. And that DNA, when they ran it, there were two bands. There's only one band, and that band was in the center. There it is in the center. It was not heavy. It was not light. No DNA at the light version. And no DNA at the heavy region. There's only DNA. 100% of the DNA was in the center. 100% of the DNA was here at the center. suggesting that it, as we said, it was either semi-conservative or it was uh, dispersive. So now we go ahead and add, and we allow that to happen again for second generation now. And now we still have bacteria here. This is a second generation. And in the second generation, we allowed it to be grown in nitrogen 14. Again, we started heavy. So we had the original strands were heavy. We only had heavy DNA, heavy nitrogen, so it was all heavy stuff. Then we allowed time to go on, the first generation, with the lighter material. And then we either had one of the two, but we had some kind of mixture of the two at that point. So if we copied it, we'd either have one heavy and one light, semi-conservative, or we'd have partially heavy and partially light. Either way, we'd get 100% one band, right? So you see this is all one long strand of new nitrogen, one long strand of the old stuff. Here it's broken up. So that would be after the first generation, which is why you ended up with 100% of the DNA in the middle, because it's either this one or it's this one. So now we let it run again with nitrogen 14. So more nitrogen 14, still light. So now what would you expect? Well, now if you ran this again, if you copied each of these again, in the semi-conservative model, you still have the old strand together, right? It's still there. You're still going to combine it with a new strand. But now the other red one is going to be used as a template, and then the second new one made of nitrogen 14 is going to be made as well. If it was truly dispersive, then you'd expect that if it was truly dispersive, then what you'd expect is that we would get two strands of the same with the same density. So this test tube, what we would expect in the test tube is semi-conservative, is we'd expect none at the light, no DNA at the lighter density is what we'd expect. We'd expect I'm sorry, I miswrote that. We'd expect that if in a semi-conservative, you see that here we have light, and then we have the middle one. We don't have any heavy because there's no nitrogen 15 to make new heavy. So we'd expect that 50%, half of this is going to be the light nitrogen. 50% is going to be the light nitrogen. And we'd expect that 50% would be in the middle. And we'd expect that none of it, we'd expect none at the heavy. There'd be no heavy DNA. That's what would be the expectation if this were true, if it were semi-conservative. If, on the other hand, it were dispersive, then as you can see here, what you'd expect is a test tube. You'd expect your test tube to have only one band, and that one band would be in the middle. We'd have 100% in the middle. We'd have none at the heavy and no DNA, and none at the light, and no DNA at the heavy. We have 100% in the middle. Well, what we actually got was 100% in the middle in the first generation. That makes sense. Either one, couldn't tell the difference. In the second generation, what we got was one light band and one middle band and nothing at the heavy. Well, that's what we would expect if it was semi-conservative. And that's in fact what we got. And this was our evidence that we needed to know that that's how we replicate DNA, by keeping the old strand, using it as a template, making a new strand. So that half the DNA is from the old stuff and half the DNA is new material. So understand that that's how we know, doing this kind of experiment with a test tube. All right, let's move on. You should be able now to answer all the questions in detail now. I hope this helps you understand how we know that DNA is the inheritable material and how it's replicated.